Okay, so I'm going to leave these two open, and you can navigate to where you need to go.
Okay. I'm going to try something crazy. Okay. You got it? Whoa, Java can handle our crappy Wi-Fi is, oh, kind of. Okay. Okay. And we are back. We are back from the break. We are refreshed. I'm so excited. Are you so excited? I'm so excited. Okay. Okay, so I wanted to demo my recently made live documentation page for the tools that I wrote to help you do analyses in Sifty-Space on your own data sets. So the package is called Siftify, GitHub Siftify, and all the code actually we're going to work through for the next section, or actually, so the code that we're going to talk about for the next section is in this running an example subject README in the documentation. And this other tutorial, working with WB View, is what we're actually going to work through today. So you can find all of the instructions again on the site. I was hoping you could all have this site open to work from as you went, but that might be a little bit difficult. So I'll try to demo this stuff as I go forward. And remember that you can completely recreate everything, every button push that I've done, I've actually recorded in this website, in this documentation page. So you can try it yourself at home. Super fun. Okay. So what we're going to be doing is building a figure that looks like this one. We'll work through the reasons why we're doing that. The first thing I want to show you, so actually the first thing we're going to do is actually the workflow that shows up in this little Siftify to your legacy data set. Hello? Hi. In a little Siftify. Okay. Hello? Hi. Hello? Oh, I'm not showing screen. Can you guys see my screen? Can you guys see my screen? Okay.
Let's turn the speaker off.
Okay. Okay. Hopefully they can speak to us. Just muted speakers. Okay. Okay, so what this does, and we'll see if we can go into Putty and do it. Okay. So what the first step that I want to show you is essentially something that exists in Siftify, a little bit of a controversial step. Just maps, say your old results or this particular map from nervousscience.org to the HCP. The HCP Connectome average surface, and we'll talk about why that might or may not be exactly what you want to do in particular cases, but it is a very fast way to build a visualization of your data on the surface. So to do that, if you happen to have a Signet account, we might not do this. Okay, we might not do this on Signet. I will show you the documentation here. Okay. So to do this, you use a command that's in my little Zipify package called Zipify Goal Result. And the trick is that it asks for a subject argument. If you give it basically the string of the word HCP as 2000 average subject, it will map it to HCP average subject. And then it takes an input file and output file, which in this case I'm using a working memory map that I downloaded from Nurses. Can you read this? So working memory FDR1, and I'm mapping it to a file with essentially just changing the extension. Just changing the code dscaler.nri. So dscaler is for descaler, and essentially format that will. Okay. So we have this. So the outputs of the transformation are actually something I put in there. I'm going to take this file I forced all of you guys to download. So you look in, if you look inside, there's a Neurosmart Maps folder, and inside that we have the original working memory nifty file, and we have that working .nri file. Now, to make any sense of this, so nifties are kind of useful in that they're already in three-dimensional space. So the information about where to actually put the data in space when you're visualizing it is already there in a file format. In a descaler file, so when we're talking about surfaces, we actually don't have that liberty. Because the truth is, with a surface, we just said that there's 6 different types of surfaces. All of those types of surfaces, so the peel surface, the white surface, the mid surface, inflated, very inflated, they all have different coordinates. So for a particular point on the surface, we might mount a map in space and make a pretty picture of. We don't actually have that information. We have different coordinates of where they should go. So we can't actually visualize any data that is on the surface without actually also loading surfaces into our visualization. So I'm going to show you how to load this data and create this pretty picture, hopefully, in Connectome Workbench. So I'm going to show you how to load this data and create this pretty picture, hopefully, in Connectome Workbench. Okay. Actually, I'm going to reopen it from scratch, just to make my life. Okay. I don't even know what that one's doing. Just to make my life. Okay. So I have. So the one nice thing about Connectome Workbench is that. Wait, I should extract. Load. Is that it's actually a tiny binary that's surprisingly easy to download unless you have the Wi-Fi that we have today in this room. Honestly. And it's actually working right now with the version that's on my key in a zip folder. So it's pretty versatile. So the part of Connectome Workbench right now is today it has 2 parts. One is called WB command, and that's a lot of Linux shell commands we can use to manipulate these files, which are awesome, and we'll get to that actually the whole part of Connectome Workbench tutorial. The other part is something called WB view, and that's the visualization. So if you open WB view, in this case we're going to open it by, I'm going to double click on it. The other part is something called WB view, and that's so if you open WB view, okay. Originally we get a page like this. So this is what happens when we open it with nothing else. So, originally we get a page like this. Okay. This is what happens when we open it with nothing else. So, this. Okay. And so the amount of buttons, and there's so many buttons. This is why we're doing this. They're all amazing buttons. They all do amazing things, but there's no buttons. And the buttons actually change. You get more buttons and more tabs depending on what type of button you have. So it kind of moves from the file. So it So what we're going to load, I might as well actually from the start. So we're going to click on Open File, and we're going to find some information load. We're going to click on Open File, and we're going to find key. Okay, so this data. So all of this data I actually pulled down from this folder is S900 average surfaces that actually I pulled down from the bulk database with an associated C. So I'm going to grab. Sorry. So I'm going to grab a couple surfaces so you can look at them. Talk about them. We can actually load multiple files at once. So we can load everything as a surface actually, because it's not too big. I'm going to load. This is the T1 data, and I'm going to load this guy, which we'll get to, which is actually so we'll load that other. And it might take a little while for this particular computer to be able to load the resolution of the T1 file. On most computers, and when you're not loading from the version of the binary that's on this runs really fast. Okay. So on this viewer, we'll try to make this big. Okay. So on this viewer at the moment, I have lots of different things to look at. So right now, plotted, at the top here, this tells you, we're in montage view. What is montage view? There's also volume view, which will show us our T1-weighted image. There's a bubble view, which shows this really cool combo thing that we can rotate. It looks really cool. And we have a viewer that's one cortex, left cortex, and right cortex. We don't need any of these. So to save us some RAM, we're going to close those windows. Okay. So these top panels show you what surface you're actually what your visual last moment. So I'm going to switch. Right now we have left mid-thickness. I'm going to switch to the left field. Notice how it doesn't. But we'll switch it to the inflated surface. You can notice it just got inflated. Okay. So this, I'm switching to left inflated. I'm going to switch this one to right inflated. You'll notice that these guys get inflated. Okay. So that gives us our inflated surfaces. That's the inflated surfaces we want to look at. Notice we don't actually need to do this, but if we unclick and click these guys, we lose and gain views. So if I want to lose all the lateral surfaces, I can unclick lateral and they disappear from this montage, or I can click them back on. Okay. Down here in this side panel, for most of you, this is at the very bottom. So this panel for these buttons will either be along the bottom or on the side here. This we call the overlay toolbox. Very fancy. So it shows you things that you can actually plot on the surface at the moment that we have loaded. So right now we have, this one's pretty cool. Notice I've changed to it and nothing happened. That's because kind of like in maybe Photoshop or something, you have to click them. You can click them on and off using the checkboxes here. Okay, so this shows you, if you've heard of Matt Glasser's myelin map excitement. I'll actually go into the myelin maps a little bit later. But we can also see, and this is the average myelin map for the 9000 release. This is average cortical thickness for the 9000 release. This is average curvature for the 9000 release, and average sulcal data for the 9000 release. Sulcus we might put at the bottom. Another thing to remember is you can, using these little arrows, you can add and remove these maps. So this guy, you can kind of add a map above or below, so you can have as many maps as you want. And you can move them up and down, and you can turn them on and off, and kind of just like Photoshop or PowerPoint, the order in which you put them is the order in which they kind of appear on the brain. So we're going to put soft-call information on the bottom, and then we're going to map, like our FMRI map on top of that. Okay. So now let's open those two files that we actually created with Ziftify. So inside Neurosynth, we have two maps here. One is the volume map, and one is the actual surface mapped data. Let's load both. And now that we've loaded our surface mapped file, we should see it here. So now we have a working memory layer that we can load. We load that. There is our surface projected working memory map. Okay, almost done. We can do fancy things, and actually I think in the tutorial I give you some fancy cool ideas, like opening this guy. So this is overlay and map settings. This lets us set our thresholds or change our color scales. So in this case, the negative values seem to be kind of weird on this histogram. Actually, pretty much, and the zeros seem to be a little bit weird. So we can click this button to remove plotting of the negative values. We can set a threshold to maybe only plot values higher than 1. So you see this little, can you see this little gray box? This shows you what's being thresholded out, or how, like the histogram of the values that are being thresholded out of your image. That probably looks good. Oh, actually, to make this representative, why don't we make, if we take this fixed map, essentially now we're saying that we want the color scale to end at 6. That's kind of weird. Maybe 8. Then we're setting the color scale to be exactly, we're setting the actual bounds of our color scale to be exactly the same. You'll see why I wanted to do that when we look at our volume. So this is our volume image. We're looking at one slice right now, but actually, might as well put this on. Yeah. So we might as well put on top of it, this is our Neurosynth map. Right now, you can see that actually by default, it didn't know what color to do, so I didn't change that. So we can open the same dialog box, and we can try to set the color scale to be exactly the same that we did in the other one. So we can get rid of negative values because they look weird, and turn thresholding on. Give it a second. Maybe we can't turn thresholding on. You can start. And importantly, right here, we have a list of all of the color scales that we can use for our maps. So I'm going to change it. I think it was on fiddle. Yeah, I'm going to change it to fiddle so that it matches the other one. And set this fixed to 8. Close. Okay, and now we have maps with the exact same color scale across both. Also, something to keep in mind with when you're looking at slices is that right now we're looking at the axial slice. You can see this little A is depressed. In the viewer, we can change this to all. And then we see all 3 slices, something similar to what you would see in FSL. Actually, my favorite that I tend to use, which might break this computer, wait for it, is to hit this button on, and it creates actually a montage of slices. So you can create multiple slices at once. Let's see. Yeah, craziness. Okay, but we can turn that off. Okay, then we can try to pick, say, whatever is the best slice that looks representative of what we're interested in looking at. Okay. Oh, the one other thing we didn't look at is, so you can barely see it, but there's this tab here. It says vol surf outline. This is quite critical. This is actually what we want. And if we click these guys on, it shows us where our peel, it shows us in green our white surface and in blue our peel surface that we've mapped to. Okay. So any question? So anything you guys notice about the surfaces that we're mapping to right now? Do they look like what you expect them to look like? It's coded at all. That's what happens when you average 1,000 surfaces together. All the places with lots of folding go away because the location of the average surface moves towards places of no folding, or at the bottom of the cell guide. But surprisingly, you might notice that the FMRI maps there relatively well too. And this is potentially actually a phenomenon to remember, because the fact that these surfaces ended up somewhere where you might not expect, it actually also applies to your data. When you do volume-based registration and volume-based smoothing, you're actually sending all of your cortical signal to these places at the same time, and you're potentially losing all of the resolution that you were getting from the folded anatomy. Okay. So one last thing, which is very exciting, is that, so you can see that I'm flipping between these panes too, these panes back and forth right now, but we might actually want to see them together. So there's actually in this view this magical button called Enter Tile Tabs. And what it does is it puts both tabs in the same window. So essentially now we have our multi-faceted figure. There is on top of this button Annotate, which tends to break on some Windows installations, so I'm guessing it will on this one too, which essentially gives you like, oh, actually it didn't break. It gives you like baby PowerPoint functionality. So we have text annotation within window. We can click here. Wait. Yeah. And we can say my figure. Okay. We can actually put the text directly on figure and save it that way. So the Essence group actually has this view that has this point of view, and hopefully it'll get adopted over time. That they actually for their own stuff, they create everything to figure, including the text, including the arrows, everything, including like the 10-page panes craziness. In this viewer, and then there's actually a spot within this viewer. We'll see if it'll work. Called Scenes. Wait, new. And see if it works. Raw, scene. Oh, wait, no. 1 second. Okay, one thing to remember, I'll come back to this later while I did that. So there's this thing called scenes, and what scenes are is this tutorial. Okay, yeah. So what scenes are is this, it's actually just an XML mark for people who like computer science. It's this file that contains all the information about all the buttons you just clicked on, essentially. And it actually contains relative paths to all the data that you are using. So the point of this, there's actually buttons here at the very top for this upload. is that you build your entire figure in here, and then you talk to Van Essen's group and get an ID to upload to, and you hit this upload button, and it sends your whole figure to their database. And we'll see, I'll download a figure. I have a figure downloaded from one of Van Essen's papers, and we'll see that when we open the figure, when we open it, it looks like the figure from the paper, but we can rotate it, we can change the layers, we can change the views, we can look at the underlying data. So it's a very powerful way to actually share your results. Okay, and the key and the weird part is that the save button isn't actually in that dialog. You need to go on this save manage files thing and see this red part which says it's not saved, and then say save checked files before you actually leave the viewer in order for that to work. Okay, so that's my, oh wait, I'm not done my 2-second demo. I have one more thing to show you. So close. One more thing that I added that I didn't add here, and this gets to a different file type. Let's go back to view from annotate. And touch bottom. Okay. There's one more file that I wanted to open and talk about, and that's a delabel file. So also available through either the Balza website or the Connectome Workbench, these things with the average surface releases, if you go get those zip files, are these things called delabel files. And these are actually surface mapped atlases that are just available for you to use right away. So with, I won't talk about that one, but I'll show you the most classic. There's a file called RSN Networks that they've been packaging with a bunch of releases for a long time. And what it actually contains, so let's move, we'll move working memory down. We'll put RSN Networks on top. So what RSN Networks actually contains, actually we'll exit File tab so we can see this better. Yeah. What RSN Networks contains are resting state network atlases that have come from the last 10 years. So this one is a classic, my favorites, the Yo7 Network Atlas. We also have in here, and you can see the name is actually printed here, which is pretty sweet, the Yo17 Network Atlas, and RSN communities from Power Neuron 2011. This is a holes-filled dilated version and a non-holes-filled projected version. There. Okay. We'll go back to Yo7 because it's the simplest. We can see it a little bit better. And the nice thing about these delabel files is that we have actually, well, one, with these delabel files, you might notice that the color of the ROIs, it's correct. It looks like the Yo map. That's because the default color is actually saved into the file somewhere. We can actually see, look through the place where it's saved into the file, and this little thingy, which I might not, okay. This little briefcase dude on the far right, if we hit it, let's go back. We open something called the Features Toolbox, and, oh, wait. It's on annotations right now. We want labels. Okay. So under labels, if we look here, we actually see the 7 network RSN labels. The magic of this little toolbox is we can now turn all of the 7 networks on and off one at a time. So this makes it this is a very useful tool for, say, browsing someone else's atlas or understanding where an ROI is. Okay, we can turn them all off. We can turn only one on. So in this case, I'm picking network 6 or the frontal parietal network because it seems to map really nicely on our working memory map. But you can see that now it's annoyingly on top of our data. We can fix that though. Check this out. So let's hide this guy so we can see it a bit better. So if we look at this, so this little wrench, this is what I clicked on to change the color scale when I had a continuous map. If we click on this wrench for a label, we get a different dialog. And it asks us, do we want to look at it filled? Do we want to look at it filled with filled and outline color, which puts a little cool black outline on the outside of it, which looks really nice. But we can also make it outline color, so just the black outline, or outline label color, so just an orange outline, or an outline matching the actual label number. Let's go with a black label outline and see what that looks like. And now we have actually a pretty cool outline of where the frontal parietal network is. And we can actually try to really gauge what the overlap of this label with that map is, which is pretty cool. Okay. Oh, and then we can save. Last step is to go back to our scene file and replace to update our figure with that outline. Cool. Okay. Okay, so all of those directions, hope that was useful. All of those directions are available on my website for you to do one step at a time if you can't remember where the button is. But hopefully that gives you an outline and basically an overview of what those things we're doing. Okay. Now let's just go back to the slides and discuss what I talked about. Okay. Okay, can the external sites see the slides? Do we know? Hopefully. Yes. Okay, great. Okay, so you might have noticed that when I talked a little bit about the file formats and how they show up differently in WB Command. One file format I didn't touch on are the time series format, so that's a format that's specific for understanding fMRI. We're going to open that in WB Command and a little bit later, actually, after we create an fMRI file that's actually in SIFT space. And the difference with that one is that, actually, we'll talk about the other two that we opened. So one that we opened was a scalar file, so our map was in a scalar file. One thing we didn't do when we created it, but we could have, is we could have actually attached the name of that map. So we could have said like neurosynth working memory map. We could have put that in the metadata of the file. And we could have concatenated multiple maps together with that label attached to that row of data. And this is actually the way that you will get, if you're working with task-based fMRI from the HCP dataset, you'll actually find these descalar maps that have within them multiple columns of data you can think of, and they all have names according to the contrast that they came from. Another type of data that you'll get, and we talked about those RSN, those resting state network atlases, are label maps, where you can see that the metadata about the label, so where it is in space, but also what number it's associated with, what name it's associated with, and what color it's associated with, are loaded into the metadata of the file, and that makes it actually show up and that makes us be able to plot it differently and work with it differently using the WB Command tools. And the last one, we'll get into time series after we've mapped some time series. Okay, so I'm saying how to actually get your data into this format. So this is something that took me a while to figure out. It was mostly me rereading a couple of modules from the HCP pipelines and rewriting them for myself to make them work on data that wasn't from HCP. And I came up with these two simple steps, which I've now wrapped in these command line tools. So the first step is you take anatomical data that you may or may not have already run through the FreeServer recon all pipeline. Once you finish the recon all pipeline, you do this siftify recon all step. So you run this command siftify recon all, and then you take the functional MRI data, which you've already pre-processed to your liking. You be you, do your thing. only two things to remember is that I want actually the T1 weighted, so not MNI transformed data, but if you have any steps that you know that you're using for bias correction, definitely do them before this step, so try to map it as well to the T1 as possible. And yeah, that is all you need to know. Then you run this SIFTify subject FMRI step, and it will combine the surfaces that we already got out of FreeSurfer with your FMRI data and give you surface mapped FMRI data. After you've completed both of these steps, there's two different QC algorithms to run, and these will generate pretty QC pages for you to work with. Okay. So the example that I'm going to work with, I don't know, yeah, you can see this pretty well. The example that I'm going to work with today is actually data that I downloaded from the open. Is the Consortium for Neuropsychiatric Genetics. Okay, so the first step to do to this data, I told you, was to convert that FreeSurfer into ReconAll. To do that, and it needs a prefix to put on your output. And that prefix should usually be unique to the type of task that you're running, so that if you have multiple tasks that you want to preprocess for the same subject, they will all get preprocessed into a folder structure that you can keep together. So in this case, my prefix is just called REST for the resting state scans. Okay. So, and a key which is important is that these REST outputs will actually get, just like with HCP, they will show up in a folder called MNI nonlinear results inside the whole HCP data structure. Okay, and then there's a SIFTiviz FMRI, very similar, which will create QC visualizations. Okay, so for a second, so I want to say that I don't think I sent it around on the key, but there is a link on my website to a download you can do, which will have this example data all downloaded, plus the QC outputs for every, for 5 subjects. So you can see how a project with 5 subjects would kind of work together. So I'm going to escape to open 2 things. One is Connectome Workbench. maybe I can. Okay. So I'll open an easier thing first. Whoa, okay. Okay. And I don't have. Wait, I don't have the QC pages. Okay. I will just open the Connectome Workbench here. Okay. So open. So once you've preprocessed data in an HCP-like way, once you've preprocessed data using either Siftify or using HCP pipelines, you tend to get these files called spec files. They're literally just text, and they give Connectome Workbench an ability to load a bunch of files at once by clicking on one thing. So if I click on the spec file, it will load all of the anatomical data related to the left-right 32K surface for this person. I'm going to list all that data, and you can say load. Oh, come on. Whoa. Okay. So this is an anatomical data file that came out of that subject, so that open FMRI data set that I ran through my pipeline. You can see now it actually does look like a real person's brain, but actually this is an MNI transformed real person's brain, so it's a real person's folding anatomy, but in the shape of the MNI surface. So if you looked at the overlap of these surfaces with, say, an MNI space mask, you would get an idea of what cortical areas were represented within those MNI coordinates for this person. And how do we set file? So we can, oh, file. Oh, so in this REST output, it's there. Oh, maybe it's not there. Oh, okay. Right. Okay, so we don't have the rest of it. I will come to it. Okay. That was from an old version that didn't work, and which we will not rewind. But I wanted to say that functional data will come in this file format called dtseries.nii. And Connectome Workbench already understands that this is a time series with a timestamp, and that these things can be used to create things like connectivity matrices. So it will actually, if you open a dtime series file, you'll actually get two links. You'll get the data itself, which you can watch in a movie, kind of surface projected, and you'll get something called a DIKON, D-I-K-O-N-N, which is actually an on-the-fly connectivity map for that person. So if you click the DIKON, you can click around in the brain, and it'll show you basically the seed correlations with the place where you clicked for any resting state file, or actually any functional MRI file, which is pretty interesting and useful. I will also say that for anyone who's interested in the futures, these clicks will work, and this will actually show you the interface for QC that I generated through here. Some of these links are broken, but they will work tonight, I swear. Okay, so back to PowerPoint. So now that we've talked about that example data, and we've talked about these file formats again, okay, I want to touch back on the actual FMRI data that exists within the HCP and how HCP have done things that are above and beyond what actually my tools are capable of. So my tools almost like cut out all of the advanced steps in order to get those pipelines to work on data that didn't have the file formats that needed those advanced steps. But now I'm going to show you what all those additional advanced steps were. So one thing to remember is that all HCP data has a high-resolution T2 weighted image. I'll talk about a little bit about that in the next slide, but it makes a big difference. The other thing is that the HCP data was collected, we already said, with 720 millisecond TR, so very short TR in a multi-band acquisition, and it was collected with 2x2x2 resolution. And additionally, everything was collected in two-phase encoding directions. And because everything was collected in two-phase encoding directions, left, right, right, left, then they can use FSL's top-up for all their distortion correction. Okay, so to compare what I talked about before with what actually exists within the HCP folders, there's some analogs here. In what I told you to do, I said just run pre-surfers recon all. Instead of running pre-surfers recon all, they run these two steps, so the pre-surfer pipeline and the post-pre-surfer pipeline. And then my little Siftify recon all is actually literally this rewritten. It's exactly the same thing. So this post-pre-surfer pipeline. I told you pre-process your FMRI any way you want in volume space, and they actually pre-process it using this FMRI volume pipeline with lots of distortion correction steps. My Siftify subject FMRI is literally a rewrite of the FMRI surface pipeline. So those things are pretty much equivalent. Okay, so this pre-surfer pipeline, the pre-surfer pipeline in the top and the pre-surfer pipeline in the bottom, you can see how they're a little bit different. Then there's a little bit more steps there, quite a bit going on than just running recon all. A lot of bias correction, a lot of T1, T2 combining stuff. This is all good stuff. And you can see how there's actually a completely different version of FreeSurfer being run, one that actually includes the T2 weighted data. This gives us two things that HCP has that most of our data sets don't. One, which is very important, is their surfaces are much nicer. So looking at this folder on the left, so the red outline here is the peel surface that you'll get out of like a vanilla FreeSurfer run, and the cyan is the surfaces they got when they brought T2 weighted data into account. So you can see all of these places where the red is going crazy and the cyan is quite nice. Also, you can see really nicely here, one of the reasons why they like T2 weighted data is that things like blood vessels show up very strongly on the T2 weighted contrast, and that doesn't exist on the T1 weighted contrast. So pretty much everything that's now adopting HCP standards, one of the golden rules is to actually have a high-res T2 so you can run these things. The other thing that they got was Matt Glasser's favorite anatomical signature, which is the myelin map, which is presented on the right. Okay. Also, this is a schematic of the pre-processing that they did before they projected to safety space. It's just there are many, many distortion correction and alignment steps, carefully thought out. And importantly, a lot of this had to do with the fact that they encoded everything in two-phase encoding directions, so they could use FSL's top-up very effectively. And what top-up does is, because it has the data in two directions, it looks for the difference between those in order to figure out what the distortion field is. So this is kind of a pre and post of distorted data versus top-up, which is quite remarkable, especially in the temporal lobes. Okay. Some last things to remember is what they did. So this kind of everything in the black box here is what they call the minimal preprocessing pipeline. There's a very elaborate neuroimage paper from, I think, 2014, which fun time to reading if you want to really understand these things. This is where most of my figures came from for this. Another thing to remember is that after they did, so they called it the minimal preprocessing pipeline because they wanted to take any steps that were essentially that anyone wouldn't potentially want in their pipeline and get rid of them, and not actually not get rid of them, but present data that was preprocessed to a level that they thought everyone would work from. So this meant that they do publish actually data that's done at this step, but then they also publish data that have been denoised with ICA-fix. So in the same full-denoised data, and be careful because the non-denoised, so the denoised data has an underscore clean. The non-denoised data does not. Also, they didn't do any filtering. They do the filtering later on. So if you want a like bandpass filter on your data, which you probably do, you're going to have to apply that afterwards. So on the clean files. I wrote here no regression movement regressions. I realized that's actually wrong because ICA-fix does remove the motion regressors. So the clean data is probably good. Remember that FMRI data in SIFTY space has the extension dTimeSeries.NII. Okay. Last thing that they did, or not last thing that they did, but one of the other major advances they did was they started throwing things other than folding into the registration. So everything is registered first according to the folding patterns of the cortex, or they call that sulcal depth. So it's called in the they use the term MSM sulc a lot in the research as a baseline to say folding that's entirely based on anatomy. It's very similar to the folding that FreeSurfer does already, but it's pretty much the same. And then they added folding, which was based on an alignment of the surfaces that's based on everything else. So they threw the myelin maps, they threw the task-related functional data, and they threw resting state ICA signals into their alignment algorithm. And they aligned all of the surfaces to try to make them as functionally realigned as possible. And they call the output of this realignment MSM all. So in the outputs, if you look at them, you'll see an underscore MSM all surfaces everywhere you can find, and those are probably the ones you actually want to work from because they're actually potentially much more anatomically aligned. And this is just a schematic of the difference that that MSML realignment made, in this case to a functional MRI signal for working memory. Okay, and the last thing, last year's Nature paper, they believe that even though that they're giving you this data in this 92K mesh, they believe that data, instead of being smoothed on that mesh, should be parcellated. Or parcellated in a way that's anatomically and functionally relevant, which is why they came forward with something like what they're calling the multimodal parcellation 1.0. So this parcellation, and this is kind of crazy. So if you look at this, this is task-related signal in the dense space, so barely smooth, but in the 92K mesh space, and this is after you parcellate. And this is the difference in the actual signal that you'll get from that. So you can actually see like a huge, in some of the critical areas, you can actually see a jump of like 10 in the Z score. It's kind of crazy. In these specific areas that seem to be related. They also claim that this is something that you can only do once you've moved to the surface, because as we've been discussing a little bit when we looked at those group averaged volumes, you actually lose the cortical spatial resolution when you do volume-based smoothing and volume-based registration to do something like this. But that's kind of a last step. I An annoying caveat is that this parcellation, while the anatomical locations of these parcels exist, this parcellation is not based on anatomy per se. It's actually a classifier based on what you expect that functional patch to look like that gets run for every person, and the output of that classifier isn't actually public yet, as far as I can tell. Okay. John, did you have anything to add or talk about?
No, I mean, I was going to demo S3 command. We do that. Okay, so
yeah.
Yeah, let's just do that really quickly, and then we can wrap up and move on.
Okay. I had some data manipulation advice, but
Oh, right, so you got more?
I got a little bit more.
Yeah, okay. Well, I'll jump in and I'll do this now, just to, seeing as we're on that demo area. And so I just need to new putty. Good. And I mean, this requires Signet to respond. Okay. So, can people see that? Is that big enough? Just a minute or two on this, really. So, reinforce the points I was making earlier. So, once you've got S3 command installed, there's a number of ways to install it. The easiest one is, well, an easy one is to use Python. You can do pip install S3 command if you have that installed. So that's one option, but I think there's plenty of others. You need to put some files in like a home folder in your, like a dot home or tilde dot, your hidden folders in your home space to tell it what your credentials are. But then, as I was saying in the slides before, get the C right. So I think this is the easiest way to browse the data, and I think if you're, if you're trying to kind of find stuff and just find out what's there, this is the most useful thing you can do. So top level, as I said before, we've got the. Okay. So you can see that. Okay. HCP 900, HCP 1200. Good question. Now I don't know what's in HCP. I think that might be like the first one. Yeah, the stuff there. So that'll be the 500 release. Probably you want to be using the 1200. To be honest, I'm not exactly sure how 1200 differs from 900, apart from that there's more stuff in. But for like one subject, I have a feeling that there's stuff in 1200 for some subjects who are in 900 that was kind of missing in 900. Because if you look through all the data, like not every subject has every single file. For as you always, obviously you'd expect some people drop out, some people get tired, fall asleep, whatever, just have a bathroom emergency in the scanner. But I think some of it is just additional analysis stuff. So there is stuff in 1200 that's not there in 900. So we've got all these subjects, and the one that probably has been the most analyzed subject in the world at this point is 10307. So you go there, you can see this is your kind of top level structure. MEG, MNI, nonlinear, that's your FMRI. T1 weighted. One or two useful things. I'll point to one or two useful things now. So in T1 weighted, this is all your this is where all your structural data is, and most of us aren't entirely comfortable with TIFT files, even when we can do anything at all with them. So you want to know where stuff you're familiar with is. So here you will find things like T1s, skull strip T1s, and if you want to know where this person's free surfer data is, it's in like kind of second, because normally in free surfer you have subjects folder and you have all of your subjects in there. So their way around making it easy to assign that in the pipelines code is they have a subject folder, but their subject folder, the subjects folder is that subjects folder /T1. So if you look in the code, that's kind of what they give that as an argument. So if you're looking for, your FS average 5 and all of these standard surf stuff, then it's there in T1 weighted, just hidden there. Also in T1 weighted is your diffusion. And there's not so much on the diffusion front. I find it quite surprising. They haven't really provided a lot of process stuff. Like this is it. This is all the diffusion data you'll get for one sub for an individual subject. Compare that to the millions of FMRI files with like several stages of processing. They've released some stuff that's like meta-analyses and group average stuff that has some tractography stuff, but there's no tractography data in any of the releases. So if you were looking for that, then hard lines, I'm afraid you have to do it yourself, or speak to someone who's run tractography on HCP. But that's where you'll find, and that's the kind of standard FSL folder structure there. Just one or two other things then. So if we kind of nosy into the, if we nosy into the FMRI, so the MNI nonlinear. Now again, there's just an epic amount of stuff in here. A lot of the stuff that Aaron was talking about, this subject's SIFTY files or this subject's structural stuff is actually in here. They're just the top level MNI nonlinear.
But actually, that's the densest, that's the biggest SIFTY files.
Right, yeah, so then it's SS average, LR, 32K. Have I done that right?
Yeah.
And then 32K stuff is in there. Yeah. And you may find yourself the real folder that you want is the data, which is in results. So here you can actually see all of the task data with the 2 phase encoding directions. There's kind of 2 for each one, and for the task data, not the rest, they have some analyses where they've concatenated the 2 phase encoding ones. So you won't want the RL, the LR if you're looking at any of these task ones, I don't think. But you don't have a combined one for the rest data. So final point, I'll just point to some MEG stuff.
So I think actually the full FSL, like contrast, are inside a combined one. So it's like FSL level 2 analysis. So you can work straight with that.
Yeah. So we have, as I said, there's these basically 3 data sets. There's resting, working memory, and the language or the story M there. And then there's this subject structural stuff. So in anatomy, you've got these SIFTY files. These are GIFTIs. These aren't SIFTIs, but these are GIFTI versions. So GIFTI is a surface format that is, in some sense, part of the nifty extension. I'm not quite sure how it fits in, but SIFTI is like a combination of nifty and GIFTI, right?
Actually, we'll go back to my table.
Okay.
But yeah.
So in the MEG, those surfaces that inflate and make thicknesses, these anatomical ones, there's GIFTI versions, and these are all there in all of the FMRI as well. You have .gii as well as the what is the full extension for the? What was it before?
Depends on what it's for.
Like mid-thickness.nii.
That one exists. So surfaces are always in .gii.
All the surfaces are
All
.gii.
surfaces are always .surf.gii.
Okay, so they're all GIFTI. All right. So here's your GIFTIs, but these are for the MEG resolution. So these are 4K. And as I told you, if you want to find the kind of standard space mesh for this, surface for this, and things like ROI labels, you need to go to the Connectome Toolbox template folder, and you'll find stuff you can do like ROI analysis for the 4K mesh. Pro tip there. It's just not obvious from anything that should be the case. Right. So last thing there. So if we look at some of the MEG stuff, like I said, we've got essentially there's 2 pipelines in the MEG. There's one that's using some ICA one uses beamforming and one uses minimum norm. And then there's two kind of fork-offs from there. And they use, and then the main kind of endpoint is those two connectivity metrics I mentioned before, the band-limited power correlations and the imaginary coherence. Step before the band-limited power correlations is the band-limited power envelopes. So this is going to give you time series for at a given frequency of the envelope, and then if you have the BF here, this is beamformer. Okay, so the beamformer source localized band-limited power correlations. There you go. They're connectivity matrices. Dense connectomes, in fact, if we have a little look, BF DLP core, yeah, so very big file names, but you can see a couple of, see here we have some .decon, so a dense connectome is going to be a correlation matrix for all vertices. So in this case, there's 4 vertices. There's 4,000 in each hemisphere. So this is an 8,000. It's 4,002. So it's 8,004 by 8,004 connectivity matrix for the decon. And then they have the pcons, which you have the same kind of thing in. Are there any decons in the FMRI?
No, because there's 20.
Because they're huge, yeah.
Huge. There's a one-liner in Connectome Workbench you can use to make them.
Yeah.
And I did it once, and then
Yep.
I regretted that decision.
Yeah. So it's kind of obvious. That'll be why they don't distribute them, because they're huge. But there's pecan. Yeah. Yeah, there's a few pecans. Pecans are parcellated connectomes. So in this case, like the Yo Atlas that Aaron was talking about earlier. So these are the kind of like highly processed connectivity you're going to be looking at in the MEG, and then the time series, like I said, in the case of the band limited power one, you've got ENV. And you'll have some DT series files. You have the PT series as well, parcellated time series, so it's kind of classic ROI time series versus this one. This will be like the biggest file. This will be 8,000 vertices times like several 100,000 data points. I think for the rest it's a couple of minutes. So these are very big files, and if you can figure out how big that is in megabytes, just by glancing at it, and you can guess. And you do labels as well, so some ROI stuff there. So just a little taste of it's really easy to just play around, explore, and then get a reasonably good idea for like what's there, and in a sense also what the different types of things are and where the different types of things go. And back to Erin.
Oh, sweet. We got a good half an hour for me to rush through. Okay. So, I wanted to do some last slides. Hopefully everyone can wake up. Everyone excited for Safety Space? Super excited? Okay, so so we have this issue that now we have, potentially from my pipeline, we have data that's on the surface, or if we're working directly from HCP, and we want to work with the Safety data, which we should. We have to understand how to actually manipulate and work with these Safety files, which is actually the second challenge, or potentially the first challenge if you're working directly from HCP. So there's some things that are really annoying when you're working with surface meshes or Safety files. Two is that one is that you can't tell from any vertex ID, so any point like within a Safety file, exactly where your data was coming from. When you load in a Nifty file, you know from where it is within the mesh, from the coordinate is in where the mesh, where that data is in space. You can't do that anymore. You need to rely on other files to do that. You do have this really great thing that things are aligned across subjects, but within a person, you have no idea what point 1000 is, really. Unless it's your favorite point, and then you can figure it out. You also can't really infer from one vertex how close you are to another vertex, because say point 1000 is probably attached to point 1001, but it could also be attached to point 3000 on the other side of the brain, because there's that third one that it needs to like loop around and also connect with. So you can't get a very good idea just based on like say 2 vertices, how far apart they are, without doing like an actual calculation that loads in the mesh. You also can't just figure out the spatial extent of your cluster from counting how many vertices you have, because those vertices are not equally spaced anymore. Usually, probably your favorite analysis script is built to work on Nifty. And that's annoying, because that's work. Okay, actually, I'm going to skip to this guy. So, if your favorite analysis script is already written in Nifty, and your favorite analysis script really doesn't actually care about the spatial information of your data or how big your clusters are, you're done, or you're almost done. So there's a nifty little function inside Connectome Workbench which will convert SIFTY data into what they call a fake Nifty. So it's data, it's a data matrix, it's wrapped in a Nifty header, time is sitting in the right spot, but all the spatial information is wrong. But if it's okay that the spatial information is wrong, because all you want to do is manipulate the data, say, in time, with no regard for the spatial information, you can work with it right away. So you can convert your data to this fake Nifty, run your magical, amazing analysis that you already know how to run, get out results in fake Nifty files, and then run a one-liner command to convert that fake Nifty back into SIFTY format so that you can visualize it and report on it. And you're done. That's awesome. Score. Only 2 extra lines to add to your code, essentially. Hopefully. Okay. If you do want to work with your file, and you do care about the surface, there's some functions, there's some main things that we normally do that exist now in FSL, which can't handle SIFTY that well, but can handle GIFTY, so geodesic format. This includes actually FSL's GLM, now can handle the GIFTY files and Nifty files, as well as FSL's PALM, which can do pretty fancy statistical permutation analyses and account for cluster size on the surface. Okay, so to run this, actually you need to use a different command to take those 3 types of data and split them into 3 files. So split them into a left cortex, a right cortex, and your subcortical data, which is now just a Nifty file. You analyze those 3 files separately using something that respects the actual 3D orientation of those data. You get out left cortex, right cortex, and subcortical result. And then you use something called SIFTY create dense time series, put them back together into a SIFTY file so that they're just packaged a little bit easier to visualize. Okay. The other thing you can do is use the command line tool, Connectome Workbench. It's pretty sweet. It actually has within it now I think 192 subcommands. So it's a one-liner, it's a WB command, and then from there you can get to a bunch of other stuff, including 192 subcommands. This includes math manipulation, statistics, parcellating, going through all these different formats. There's just a ton in there, and actually the actual doc strings in those functions, John doesn't like them, but I kind of love them. I find them pretty. There's no examples, but they're pretty intuitive, and if you use them wrong, they tell you. So if you forget a flag, they'll be like, you forgot this flag. And you can usually figure them out relatively well, in some cases, for some straightforward things. So there's things like math, averaging, reducing, merging. I don't think I have merging. So concatenating multiple files together, doing weighted stats, doing regular stats, smoothing on the surface, or smoothing actually in a anatomically specific way. So 2D on the surface, 3D in the volume. Finding clusters, which is kind of like FSL's clusterize. Correlate, which creates those DICON files, so those huge connectivity matrix. Oh, and this one, very cool, file information. So W command file information, it's like FSL info on your crack. It prints to the screen a whole lot of information about what is in your SIFTY file and how it's organized, and what the data looks like. Okay, but like John complained that sometimes there's no examples, and sometimes it can be hard to figure out how to use this, and sometimes you actually need to chain 4 of these functions together to actually do what feels like one thing. So in those cases, as I've been going through, I've been basically documenting the way that I do things and wrapping, say, 4 commands together into these little command line tools that exist in the SIFT package. I also sometimes hook into Python's nibabble to do some extra little things. So one thing is SIFT define mean TS, which can essentially take any file format and give it a functional file or a scalar file, and will just output the mean values or weighted mean values according to some masks. There's SIFTify seed core, which creates seed correlation maps in a very similar way. There's something called SIFTify surface ROIs, and that basically just reads in a lot of coordinates or a lot of values and creates circular ROIs on the surface, so you can go ahead with analyses. And there's something called SIFTify peak table, which is essentially my little rewrite of what FSL clusterize tends to do. So for this particular fate cluster that I created in the precuneus, if you feed it into peak table, it'll output this CSV with information about what hemisphere it is, what the peak vertex is, what the size of the cluster is at a particular threshold. And it will also output, so you can't really see in blue, but you can kind of see in white, this is the cluster that it got from that. This is the cluster that it defined at the threshold I gave it. And it will actually tell you about how your cluster overlaps with certain atlases as well. So it'll tell you how it overlaps with the FreeSurfer atlas. You can get an anatomical label. In this case, we say that it's 60% overlap with the left precuneus label from FreeSurfer. It'll tell you that this particular fate cluster I created is actually entirely encompassed within the default mode network from the Yo atlas, and that 30% of it and most of it is actually overlapping with L31PV from the new Glasser MMPI atlas. So that is essentially where I wanted to stop. Other things to know is that nibabble can handle these file formats. That's what I use. And MATLAB's field trip.
Yeah, so field trip's got some sort of reading functions. Maybe that's not for it, I'm sure.
Yeah.
You definitely do stuff in MATLAB. That's probably the first of all. Python nibabble support has been like in depth for a long time, but it's come out the latest release in the last maybe 2 months. So it's now officially you can read CFP in nibabble, but I don't think you can get all of the information from the files. At least it's kind of hidden away in weird classes that you don't need to touch.
Yeah.
So
although I have kind of figured it out.
Entirely supported. And to get the numbers out, I think there's additional stuff.
Yeah, a lot of stuff in there. And I tend to, so another thing is that these command line tools, there's some Python functions that they rely on. That's all also in the package that you can read through. And when it uses nibabble, actually what I do is I actually separate the files and I read them as GIFTs. I read the data from GIFT format than actually from the SIFT format because I find it actually a little bit more reliable. There's this implicit masking that happens for the inside of the brain, and it's not exactly the same mask in every person. It can cause alignment problems. It's a weird caveat. Any more questions? Interesting things? We had a lot of information today.
Sure, this is a good question.
Yeah.
For your safety package,
yeah.
You run under like a Python environment or under?
Yeah. Yeah. So it's under it's a Python environment, so it can be and load. It will go up on Pippi soon. Right now, it can be installed from the GitHub release using pip. Yes, and there are versions of it that are already installed on things like the Signet cluster that people can work from right away.
But it's command line, so
Yeah, and I do plan. It's actually and most of the functionality is actually command line tools. So once you have the Python environment kind of loaded, then it would work from the command line like anything else, and you wouldn't really need to know Python to be able to interact with it. Hopefully in the near future too, I'll wrap it in a Docker so that it'll be easier to install on different things. It does potentially, actually. So first,
better you use like a Mac or Linux instead of Windows.
Yeah. Well, we'll see. If I wrap it in the Docker would also contain FSL and Connectome Workbench, because in order to get it to work. There's also one function that gets called from the FreeSurfer library, and I'm trying to figure out if I can get around that without having to put another 10 gigs inside the Docker image. Yeah, literally just MRI convert.
That's like half reset.
Yeah. Yes.
Another question about
I was going to ask if these slides can be available, particularly the one about on the course website.
Yes.
And we can get started.
Yeah. Oh, so yeah, just Google like HCP summer course. Those are all there. These slides can be made available, but pretty much all the information on these slides are also in the SIFTify documentation webpage as well, hopefully. If not, it needs to go there in the near future. Yeah.
For surface analysis, what's your recommendation? You can go back to the HCP Workbench route, and you can do like you said, you have 3 step, 1 and 3.
Yeah.
Which one do you think is more convenient?
It depends.
All
Yeah.
probably already have some procedure to do our analysis.
Yeah.
Then we just want to add the surface part. So which way?
So if you don't need your so if your spatial information isn't a part of your analysis, then using the fake Nifty is by far the fastest and easiest to work from. Connectome Workbench as a toolkit, like the WB command, is C-compiled libraries that are definitely like probably the most efficient and fastest if you want to do something fancier with the data. And there's actually a lot of subcommands if you actually read through the documentation that I'm like, I didn't know I could do that. Now I do. I'm so excited. So it is useful to learn.
There's some things, particularly with very, well, specifically with very huge files that you might want to consider using WB command for, because it's probably the most optimized tool and maybe the only one that's capable of dealing with 30 gigabyte files.
Yeah.
For certain kind of operations.
Yeah, like
PCA and that is
yeah, even like time series extraction. I'd originally written the whole thing in Python, and then I switched to, in most cases, actually just using SIFTI parcelate or W command parcelate, and it ran like 100 times faster. It was just ridiculous. Maybe I was using terrible Python.
I'm sitting down with a new data set, and I want to throw out my old analysis, and I want to be the cool new kid.
Yeah.
Old analysis. Do I do it all with WB command?
Potentially.
You're asking an old man.
Yeah.
Yes, I think
I mostly do.
the answer is partly yes.
Because
Partly
like,
yes.
a few years ago, the answer would have been pretty simple, right? I wanted to do
Yeah.
So it kind of does an awful lot of good, so it seems like a kind of good candidate replacement for these. I don't know if that's
Yeah.
intended or not.
A little bit. Like, I really appreciate the fact that the WB command version is already contralateral. It makes my life a lot easier. And the NIBabel reading of the GIFTY files, actually, I prefer it. Actually, I didn't start doing anything super sophisticated in FreeSurfer, so I'm maybe not the right person to ask if you already know FreeSurfer really, really well. But if you need to learn something from scratch, I would start here instead.
But actually, I mean, I think a better answer to the question is, if you want to be the cool kid with cool tools, then just be familiar with SIFTI, and WB command is currently the best tool for working with SIFTI.
Yeah.
But in 5 years, like, they'll probably be lost.
Yeah.
So then it might not be the best. But if you're familiar with SIFTI, then you probably
Yeah. I should say also that I meant to say, but I didn't say, but all of the tools that I write, if you run them with like the debug flag, it'll actually print all the commands. So you can get like example usage of what I do in SIFTI and WB command by just like running one subject and printing out the commands and deciding what you want to do from there.
Okay, well, if there's no other questions, I want this workshop. Five words at the start. Thanks a lot, Aaron. Really appreciate you coming along. And thanks for coming, guys. You know, any general questions, just feel free to email with us. We'll help you out with your problems and your questions. Good luck.
Thank you. You've been awesome. Absolutely great. And you helped out. Well, completely solved like some pretty serious software problems, which I'm very sorry about that.
No, like, you got to be prepared. I wasn't anticipating something that was being webcasted from somewhere with no Wi-Fi.
Yeah, well, to be honest, like, I've been in this room before, and the Wi-Fi is worse today than I've ever seen it.
Yeah.
So I didn't quite plan for that
Yeah.
eventuality. Thank you.
Yes.
I just wonder if there's going to be a turn of brain
Yes, and maybe not in March.
Okay.
Yeah. That's because, yeah, last year we planned it with BrainHack Global, which hasn't been announced yet. Like, there hasn't been like a true BrainHack Global announced, so I don't know if we're going to talk to the BrainHack people about whether or not we'll do it wrong one.
Oh.
Maybe we can send them out.
Yeah, but hopefully,
Jen has all the emails.
Yeah.
Right?
Yeah.
So we'll send them out.
Sorry about that.
How'd everything else go? I was listening in.
It went great. I think the kind of many improvisations that came along, like, solved a pretty severe potential technical problem.
Yeah. So this is
We never do a workshop in this room again.
Yeah, it's
Ever.
over Wi-Fi, right? Yeah. Have you been in better rooms? Are there better rooms to do them in?
Well, the 7 whatever, the main meeting room, like that one's generally okay.
748?
Yeah.
Oh, God, that's good.
It's just the Wi-Fi on the floor is just like everywhere is rubbish.
Yeah.
All over the floor.
There's, I don't know if Jen's talked to Al about this before.
There must be.
Because 748 would have worked fine for this one, too.
Oh, right. Well,
Right? Yeah.
I thought it was booked. That's why we're in here.
This one is booked for this one. I don't know if Jen also books it.
Ending webinar. Ending webinar, yeah?
Yeah.